Welcome everybody to today's video. In today's video, I'm going to be talking about protein synthesis, or specifically the main ideas that you need to know in order to be ready to go deep into details. In order to understand the process, we need to understand what is protein. What is protein made up of? Okay, I can tell you that protein is made up of polypeptide chains, like this in the image here. This is a polypeptide chain that is made up of amino acids bonded together by peptide bonds. And these amino acids are the translation of the DNA codes, okay, in the cytoplasm, because we have the cell and we have the nucleus. Here we have the DNA. Okay, it's a simple drawing. The DNA has codes. These codes, okay, will code for a protein. How? By coding to amino acids. And these amino acids will bond together by peptide bond to form a protein. How does the DNA strand code for these amino acids? This process is the process that we're gonna explain in part two and part three, and we're gonna give like main ideas in this part one. Okay, so the DNA has codes and need to transfer these codes to the cytoplasm where they are gonna be translated. Okay, how is this process gonna happen? How are codes gonna be transferred to the cytoplasm by the mRNA? It will transfer to the cytoplasm, and then by the tRNA and a kind of help from the rRNA, it will be translated to these amino acids. And this process, I'll explain it in details in the later parts. And this image illustrates how the polypeptide chain, which is a result of the translation of the codes of the DNA, okay, is the unit of the protein, is the main unit. It's the primary structure, polypeptide. These are amino acids with peptide bonds between them to form the polypeptide. And the polypeptide will become a helix. It may coil and join with a protein to form tertiary structure, quaternary structure, and then to form the protein. And now we know, since most polypeptides are manufactured at ribosomes in the cytoplasm, okay, and the DNA never leaves the nucleus, we know that the DNA requires not only a decoder of the codes, but also a messenger as well. Okay? Here we have three types of RNA: mRNA, tRNA, rRNA. The mRNA is the messenger, the tRNA is the transfer RNA, rRNA is the ribosomal RNA. These are the names, okay? So the decoding and messenger functions are carried out by RNA, which is the second type of nucleic acid. The first type is DNA, we know that. So these three forms of RNA typically act together to carry out DNA's instructions for polypeptide synthesis and then proteins. Okay? And beginning with the messenger RNA, which is a relatively long nucleotide strand like this one, that we can notice it resembles one of the DNA strands because the DNA is something like this. If we take only one strand, it will be like the RNA. It's only one strand that has nucleotides. mRNA carries the DNA codes to the cytoplasm. And then after this transmission, we have the decoding process, which is the translation. And here we have the transfer RNA. These molecules ferry amino acids to the ribosomes. So here, in this location, amino acid at the attachment site, we have an amino acid, and this transfer the RNA will bring this amino acid, will transfer it to the ribosomes to be part of the transcription process, which we'll talk about it in the part three. And when they arrive to this site, to the ribosomes, They decode mRNA's message. Okay, so this anticodon should be appropriate to this codon. And this is another story, GAGCUC. It's a really simple one. I can explain it, but now we are focusing on the process, okay? On the main ideas in the processes. So I'm first focusing on the mRNAs just to know what each one will do in the following parts. Okay, so then the amino acid sequence, the polypeptide will be built. And the last type of RNAs is the rRNA, the ribosomal RNA. Don't bother yourself with the structure, it's really complicated. And I'll just tell you the important things that you need to know. These rRNAs form the ribosomes. What are the ribosomes? The ribosomes are basically two subunits, large one and small one, like this. We can say it's the place that where translation occurs. So here we have the translation. This is the large subunit. This is the small subunit of the ribosomes. And these ribosomes consist of proteins as well as rRNAs. So now we know that the mRNA, this one, is the one that will, it's a messenger. Okay, so it will take the codes from the DNA and go through these pores, nuclear pores, to the cytoplasm, to the ribosomes. And here, the translation will occur. Here in the nucleus, the transcription will occur, and here, the translation, where a polypeptide chain is formed after the involvement, of course, of, here we have the ribosomes, if we go. This is the large subunit, the small subunit. The tRNA will be like this. This is the proper shape of it. I will not go into details of its shape, okay? And the mRNA will be here. And as we said that the ribosomal RNA will form the ribosomes along with the proteins. Thanks everybody for watching this video. This was part 1 and part 2 will be about the transcription. So don't forget to hit the like button and subscribe for more. See you in the other video.